First, adrenaline is synthesized inside the interneuron and stored in synaptic vesicles at the axon terminal. When an action potential reaches the interneuron's terminal, it triggers the opening of voltage-gated calcium channels. The influx of calcium ions causes the vesicles containing adrenaline to fuse with the cell membrane. Adrenaline is released into the synaptic cleft between the interneuron and motor neuron by exocytosis. Adrenaline binds to adrenaline-gated sodium channels on the motor neuron, initiating the excitatory response. Adrenaline binds to adrenaline-gated sodium channels on the dendrites of the motor neuron. These channels open, allowing an influx of sodium ions into the motor neuron. The increased positive change depolarizes the dendritic membrane, creating an excitatory postsynaptic potential, or EPSP. Once the EPSP is strong enough to reach the threshold potential, voltage-gated sodium channels at the axon hillock open. Sodium ions rush in, further depolarizing the membrane. This rapid depolarization triggers the firing of an action potential. As the membrane becomes highly positive, voltage-gated potassium channels open, allowing potassium ions to flow out, repolarizing the membrane. The sodium-potassium pump restores the ion gradient by pumping 3 sodium ions out and 2 potassium for every 2 potassiums in. The action potential travels down the axon as sequential opening and closing of the voltage-gated sodium and potassium channels propagate the signal. This wave-like transmission ensures that the signal reaches the axon terminal. When the action potential reaches the axon terminal, it triggers the opening of voltage-gated calcium channels. These calcium ions flood into the terminal, binding to the vesicles, causing synaptic vesicles containing acetylcholine neurotransmitter to fuse with the membrane. Acetylcholine is released into the synaptic cleft by exocytosis. Acetylcholine then binds to acetylcholine-gated sodium channels on the muscle cell membrane. These channels open, allowing sodium ions to rush in, depolarizing the muscle membrane. This triggers an action potential in the muscle cell, which spreads across the sarcolemma and down T-tubules. The action potential stimulates the release of calcium ions from the sarcoplasmic reticulum inside the muscle cell. Calcium ions initiate the contraction by allowing actin and myosin filaments to slide past each other. This leads to muscle contraction, which enables the clam to dig. Saxitoxin, a potent neurotoxin, blocks voltage-gated sodium channels on nerve and muscle cells by binding tightly to the channel's outer pore. This prevents the influx of sodium ions required to generate action potentials, effectively halting nerve signal transmission. In clams, this disruption presents muscle contraction, paralyzing their movement and interfering with essential functions such as shell opening and closing. Saxitoxin is released during harmful algal blooms, commonly referred to as red tides. These blooms occur when dinoflagellates proliferate rapidly, often due to nutrient pollution and favorable environmental conditions. The reddish hue of the water is caused by the high concentration of pigmented dinoflagellates. As they grow and die, they release saxitoxin into the surrounding water, where it accumulates in filter-feeding organisms like clams, leading to paralytic shellfish poisoning risks for other marine life and humans.